Hello students, so once again I welcome you on our YouTube channel Little Kingdom School. So we are going to discuss some MCQ based on the topic which we are already learned in previous video. So today I insist you to keep your NCERT with you, keep your notebook and pen with you to mention these MCQs and how to solve this MCQ. Done? So let's start. The first MCQ. How many meiosis is required for the formation of 400 pollen grain? So this MCQ is based on the topic of microsporogenesis and the pollen grain. So let's discuss or let's revise this topic first and then we'll move towards the MCQ. Let's see. As we know, in each anther there are 4 theca and in each theca contain microsporangia, and microsporangia contained 4 layer structure. Outermost is epidermis, then endothecium, then middle layer and then tapetum, and in the innermost side the sporogenous tissue is present. So right here I am drawing a sporogenous tissue, clear, which is diploid in ploidy. Next, this sporogenous tissue undergo differentiation. After differentiation, it convert into pollen mother cell, short form PMC, which is also diploid because there is no meiosis. Next, this pollen mother cell undergo meiosis. This pollen mother cell undergo meiosis to produce 4 haploid microspore. We know already at the end of meiosis 4 daughter cell produced. Each cell contain half the number of chromosome. So 4 haploid cell produced called microspore, and all 4 cell are remain attached with each other, and the structure is called microspore tetrad. So microspore tetrad, which is haploid. Why? Because meiosis take place in pollen mother cell. Microspore tetrad after some time different from each other, that means each and every cell are separate from each other, and now they are known as microspore, which is haploid, and each and every microspore in future mature into the pollen grain. And as we know, pollen grain contain 2 layers, inner layer intine, outermost layer exine, and 2 cells. Larger one is vegetative cell and the smaller one is a generative cell. We can say that from the meiosis of one pollen mother cell, total 4 microspore produced, and from this 4 microspore after maturation, total 4 pollen grain will produce. So this structure is pollen grain. Pollen grain, which is also haploid. How many pollen grain is going to produced from the single pollen mother cell? 4. So we can write that from single pollen mother cell we get 4 pollen grain at the end. Now let's move towards the MCQ. How many meiosis is required for the formation of 400 pollen grain? That means we need to form 400 pollen grain. Now we know that as a result of one meiosis we get 4 pollen grain at the end, and we need to produce 400 pollen grain, then it required 100 meiosis. Clear? So the answer is 100. Why? We know that the core concept is one meiosis produced 4 pollen grain. That means simply 100 meiosis produced 400 pollen grain. So the answer is 100. Next MCQ, second MCQ. How many pollen mother cell are required to form 1200 pollen grain? The concept is same, but instead of meiosis, there is pollen mother cell. That means PMC. Again, let's see this concept. Now, one pollen mother cell undergo meiosis to produce 4 pollen grain. That means 4 pollen grain is produced from one pollen mother cell. We need to produce 1200 pollen grain. Simple answer. You know the answer. What is the core concept? One pollen mother cell produced 4 pollen grain. Then 300 pollen mother cell produced 1200 pollen grain. So the answer is 300. This type of MCQ is frequently asked in your board examination, good set examination, as well as in your NEET examination. Now next, how many pollen mother cell present in each sac of anther? If mature anther contains 1600 pollen grain, this one is quite tricky MCQ. Let's solve this MCQ. First of all, if mature anther contain 600 pollen grain, then that means there are total 400 pollen mother cell. Let's write from the beginning. Simple. One pollen mother cell produced 4 pollen grain. That means total 400 pollen mother cell produced 1600 pollen grain. But the answer is not 400 pollen mother cell, because the question is how many pollen mother cell present in each sac. Each sac is the key word. We know that each anther contains 4 sac. If you want to remember the diagram, it is like this. And it is a thin section or transverse section of anther, bilobed anther. You can easily observe all 4 theca or all 4 pollen sac or all 4 microsporangia in this structure. 1, 2, 3 and 4. Now the answer is simple. If the anther contain 400 pollen mother cell, due to which it produces 1600 pollen grain, and the question is how many pollen mother cell in each sac. We have total 4 sac. So if anther contain 400 pollen mother cell, then each sac of anther contain only 100 pollen mother cell. 100 in this sac, 100 in this sac, 100 in this sac and 100 in this sac. That means 100, 200, 300, 400. Total 400 pollen mother cell present, but in each sac total 100 pollen mother cell is present. So the answer is 100. Next, how many pollen grain form from 120 pollen mother cell? In this type of MCQ, we need to find out the number of pollen mother cell or number of meiosis. But in this case, the number of pollen mother cell is already given. We need to find out how many pollen grain is formed from 125 pollen mother cell. Core concept is again same. If one pollen mother cell produced 4 pollen grain, then from 125 pollen mother cell we got 500 pollen grain. So the answer is 500. Clear? So on the basis of microsporogenesis and the pollen grain, this type of MCQ is frequently asked. Now next, the sum of the topic which is given in your NCERT, like pollen allergy, pollen viability, pollen product, which is not too much difficult to remember. So I will discuss it here without board work because it does not need any board work. So the first is pollen allergy. What is pollen allergy? So some pollen grain are able to cause allergy in our body, and able to cause some serious disease like bronchitis and asthma. Clear? So we need to take care of some kind of pollen grain. So वो कोई ना कोई तरीके से हमें एलर्जी कर सकती है, बट एग्जाम्पल टेक्स्ट बुक में मेंशन किया गया है विच इज पार्थेनियम और द कैरोट ग्रास. द स्पेसिफिक प्लांट इज पार्थेनियम और कैरोट ग्रास which is come with a wheat species. Wheat species is exported from a foreign country in India, and with the wheat species some plants of parthenium or carrot grass come or enter into the India, and now it produced allergy. It caused allergy to many people, and it caused serious disease like bronchitis and asthma. Clear? So it is about pollen allergy. Next one is pollen product. So pollen grain is also nutrition rich material or nutrition rich compound. उसके अंदर काफ़ी सारा nutrition है. So it is also used as a product in some western country. Pollen tablet is there, pollen syrup is there, which is frequently used by the athletes and the race horse to increase the performance. Next one is pollen viability. Now what is pollen viability? Let's understand. For example, if pollen grain is produced in anther and once it is released from the anther, then how many time it's remain viable? वो कितना time viable रहेगा उसको बोलेंगे pollen viability. So in your textbook there are two examples. In cereals like wheat and maize, the pollen grain remain viable for approximately 30 minutes, and in some plant family like Solanaceae, Rosaceae, it remain viable for up to a certain months. We can also store the pollen grain with the technique of cryopreservation, which use liquid nitrogen at minus 196 degree centigrade. Clear? So this is all about the pollen viability, pollen product and pollen allergy. Now we are moving towards the next topic, and our next topic is female reproductive structure. As we know that the female reproductive structure is pistil or gynoecium. How it's look like? We already draw the diagram in very first video. So it look like this. It's look like this. Then it contains three parts. First, stigma. Second, style. And third one is ovary. So the female reproductive part totally contains three parts: stigma, style and ovary. What is stigma? So stigma provide landing site for pollen grain. That means from the anther pollen grain is released and it come on the surface of stigma with the help of some pollinating agent like wind, water and insects. Then ovary is a basal part of female reproductive structure. And what is style? Style connect the stigma and ovary. It connect the stigma and ovary. Based on the number of pistil, the flower are divided into two types. Let's discuss this. And one more thing, this pistil is also called carpel. We can also call this carpel. Then, so we are using this word to divide flower on the based on gynoecium. So the types of flower, based on number of carpel, that means number of pistil. If flower contain only one pistil, then this type of flower is called monocarpellary flower. And flower contain more than one pistil, then it is called multicarpellary flower. If flower contain only one pistil, so there are not further division, but if flower contain more than one pistil or more than one carpel, it is further divided into a two type. First, apocarpus, and second, syncarpus. Syncarpus. If there are more than one carpel and all the carpel are free from each other, they are not fused, then it is known as apocarpus flower. And the example of apocarpus flower is Michelia. Michelia. It is the example. And if there are more than one carpel present and all the carpel are fused with each other, they are not free, then this kind of flower is called syncarpus flower. And the example for syncarpus flower is papaver. Papaver. These two are the most common example. Diagram is given in your NCERT and the MCQ is frequently asked based on the diagram of Michelia and papaver. How many types of or the types of carpel present in Michelia? So we should know that multicarpellary apocarpus, the type of carpel or type of flower present in papaver, then it is multicarpellary syncarpus. Frequently asked MCQ. Next, after this, we need to learn what is Megasporangium. This is all about the structure of pistil and the type of flower based on the number of pistil. Now, Megasporangium. Megasporangium, the most important topic for board perspective as well as for MCQ perspective. What is Megasporangium? So before starting Megasporangium, we should know the basic structure of pistil, which look like this. I am not drawing it again. You know that stigma, style, and ovary. Inside the ovary, Megasporangium is present. So for your reference, I am going to draw this wall of ovary. Otherwise, it is okay if I will not draw. The board will not cut the mark for this. So I draw the basal wall of ovary for your reference point. Suppose wall of ovary, I draw only this part from this to this. Then zoom करके draw किया है. So it is wall of ovary. It contains placenta. Yes, placenta is also present in plants. It is not only present in animal, placenta is also present in plant. On this placenta, the structure of Megasporangium is present like this. So it is the structure of Megasporangium. The structure of Megasporangium is also called structure of ovule, and it is also called a typical anatropous ovule. This Megasporangium, these ovule are of multiple types: anatropous ovule, amphitropous ovule, hemitropous ovule, cincinotropous ovule. But in our syllabus, there is only one type of ovule, which is anatropous ovule. Anatropous ovule. So question can be asked like this: draw the diagram of typical anatropous ovule. So we need to draw diagram of this type of ovule. So this one is ovary wall, placental tissue, placental tissue which provide nutrition to this megasporangium. Now, megasporangium has a stalk-like structure by which it is going to attach with placenta. So this one is stalk-like structure known as funicle. It is known as funicle. What is funicle? Stalk-like structure by which ovule is attached with placenta. Next, this one is body of ovule. This upper portion is known as body of ovule. This lower portion is stalk of ovule. So there is a junction point which connect the both funicle and the body of ovule, and this one is the junction point, is known as hilum. So hilum is the junction point of this funicle and the body of ovule. So it is known as hilum. Hilum is a junction point of funicle and body of ovule. Then inside this ovule, this empty space, I will highlight it for you. This empty space contain a specific nutritional tissue known as nucellus. The function of nucellus we'll discuss quite later. And in the center of nucellus, the most important structure of ovule is present, called embryo sac. What is embryo sac? We're going to discuss in the next topic, but right now we need to discuss up to nucellus. This embryo sac is most important for the further development, for the reproduction. So the tissues of nucellus provide nutrition to this embryo sac. Nucellus provide nutrition to embryo sac. As we know, embryo sac is the most important structure, so it is protected by two-layered wall. This one, first wall and second wall, and this wall or covering of this ovule is called integuments. So the outer integuments, which is outer covering, and the inner integuments, which is inner covering, Now if you observe this structure carefully, we got two ends. The end with integuments. On this end, both the integuments are present, and the end without integuments. So the end with integument, that means this end, is called chalazal end. We are going to use this end, this chalazal end and this end for the further topic of reproduction, in the next topic of reproduction. And this end without integuments is called micropyle end. So the most important diagram, most important topic, it can be asked for 4 marks in board examination. Only diagram can be asked for 2 marks in your board examination, and it can be asked without diagram. You have to write only theory, which I mentioned here, for 3 marks. So let's revise it again. Megasporangium, also called ovule, also known as typical anatropous ovule. So it is present inside the ovary, swollen basal part of pistil called ovary. The wall of ovary, it contains nutritional tissue called placenta, which provide nutrition to this ovule. So first part, funicle. This one is funicle. Funicle is a stalk-like structure which help to ovule to attach with this placenta. Clear? So funicle. Then next, hilum. Hilum is the junction point between the funicle and the body of ovule. So this one is the junction point called hilum. Then tissues of nucellus. This one. Tissues of nucellus is a highly neutralized tissue which contain so much amount of nutrition and which help in the development of embryo sac. Embryo sac is a most important structure for reproduction. So this embryo sac is well protected by two layer covering. Outer layer is called outer integuments and inner layer is called inner integuments. So outer covering and inner covering. Apart from this, the end with integuments is called chalazal end and the end without integuments is called micropyle end. Now don't be confused by looking this diagram in your NCERT. This diagram is drawn in upright position like this. Right? So I draw in the straight position. So nothing wrong in this diagram. Clear? So this is all about the Megasporangium. Thank you.